So funny that it says that now. Okay, go for it, Jess.
Hi, everyone. Good morning, good morning, good morning, or good afternoon, based on where you are. So thank you for joining us this morning for the lightning talks. So we just want to start with giving you a little bit of information of how this session will be run. So each lightning talk presenter will present for about 10 minutes. And we have 3 this morning, so they will go one right after the other. After the presentations are done, we ask that you hold any questions or insights or thoughts until the very end. What we will do is that we will open up the breakout rooms, and at that point, you will have the ability to choose which breakout rooms you want to go to. So you can actually navigate between them throughout the next hour. And then at 10:30 a.m., we will have a hard stop to make sure that everyone has at least a 30-minute break before we get started again at the top of the hour. But however, if you want to stay in those rooms for a bit longer because the conversation is that engaging, then you can, but we will send you a notification saying, hey, 10:30, you might want to a.m. Eastern time, you may want to take a break before we get started at the top of the hour. For our presenters, just a quick reminder, you do have 10 minutes and we will shower you with love. And so you will see little hearts on your screen to let you know that you have 1 minute before your time is ending. I think that's about it. Donalyn, is there anything else I'm missing?
I think that was great. Any questions? Oh, I think you're good to go.
Okay, awesome. So Dongheon, are you ready?
Yeah, sure. I can go ahead. Yeah.
Okay, so whenever you're ready, the floor is yours.
Thank you. So hello, everyone. It's good to present my work in today's conference. So today I will present one of my last PhD work. Last project I completed when I was in PhD student in Texas A&M University. So today I will briefly talk about the one way to construct a biological system model when we have a limited knowledge of a system. So we want to use something called hybrid modeling approach to construct the mathematical model for many of the uncertain signaling pathway as well as the biological system in general. So the particular system I'm interested in during my PhD study is basically cellular signaling pathway. For many of the audience in here probably know what cellular signaling pathway. So cellular signaling pathway is basically how the cell process and process as well as how receive and process and respond to the external perturbation received from its microenvironment. One example I'm showing here, NF-kappa-based signal pathway, which I have studied and modeled throughout my PhD study. And signal pathway looks very complicated, but overall, in general, signal pathway can compose to 3 steps. First one is when perturbation activates the cellular receptor on the cytoplasm, and the activated receptor will activate a series of protein activity change, which in the end cause the change in gene expression. This change in gene expression will help the cell properly address the problem arising in microenvironment. So in order to understand the cellular signal pathway, usually a combination of both modeling and experimentation are implemented alongside each other so that we can study the complex interaction among the other biomolecules involved in the process. So mathematical models are important in the sense that we want to generalize the hypothesis in proposing literature as well as trying to interpret the data from experimentation. However, the problem usually lies when we try to formulate the model. So because the system is quite complex and usually our prior knowledge of the system is quite limited. So in the end, the formula this is why the formulated model based on our current understanding can be quite limiting. So this example we're showing here. So based on suppose that our knowledge of the system is quite limited, then the formulated model can be can be inaccurate compared to the true system. Under such circumstance, the model prediction accuracy can be limiting, and it cannot be accurate used for analyze the system we are interested. under this scenario, what we propose is we're trying to combine a mechanistic model as well as data-driven model. So mechanistic model is something we usually use for underlying the bio system. So suppose that we trying to use the ordinary differential equation, then we trying to use the ordinary differential equation model to capture our current understanding, where X is the concentration of biomolecule, such as protein and mRNA, and theta will be captured the parameter to characterize the interaction among all the biomolecule. So the mechanism model is useful when you have it's useful because it incorporates the underlying mechanism and it has the interpretability. On the other hand, data-driven model is inferred directly from experiment with more reliable accuracy. So this is how it works. So we have a multiple different perturbation condition as well as we have measurement. Then we try to directly mapping this perturbation condition with the measurement we have. So it can be tailored to the data set we have. However, it's more like a black box model. We don't know what's actually causing the measurement to be so under particular condition. So in this study, we trying to combine this mechanistic model as data-driven model. So in a hybrid model, we formulate as such. So first, we formulate the mechanistic model based on prior knowledge. So this is the ordinary differential equation model. And trying to modify the mechanistic model by adding W, which is inferred from experiment. So that we can, the accuracy of the overall hybrid model is enhanced. So the schematic illustration is showing here. So here, if you're on the left, original hybrid model cannot predict the original model prediction, showing the dashed line cannot match well with experiment measurement even after parameter estimation. By adding W inferred from experiment, we can actually increase the accuracy of a model prediction, but to match with experiment measurement better. So based on this, the overall procedure for constructing hybrid model is following. First, construct a mechanistic model. And next one, you estimate W for measurement. And last one, you're trying to construct the empirical mapping between the original model with estimate W so that we can generalize our prediction accuracy. So I will briefly go over how we construct a model. So suppose we have a mechanistic model available and we have an experiment measurement here. So what we want to do is trying to solve the conventional parameter estimation problem here, where objective function is trying to minimize difference between hybrid model and the experiment measurement. And the decision variable with W, where we trying to estimate W at each time point where measurements are available. By estimating W now and including the hybrid model formulation, we can actually increase the model prediction by adding W. So next step will be trying to generalize this hybrid model prediction. So one limitation of a previous approach is that we need to have a W in order to have an accurate model prediction. But however, W is only available when you have measurement. This sometimes can be problematic because you want to employ the mathematical model to predict the system dynamic under new condition. However, under new condition, there are no measurement, which means there are no W. So under this case, you can the implementation of the hybrid model can be limited. So what we want to do is actually we trying to find the empirical mapping between the original ordinary differential equation and the estimate W from available measurement. So that in this sense, then we only need to simulate the original hybrid model, original ordinary differential equation model, so that we can predict the W under any condition we want. So here we use the artificial neural network because it has proven its ability to approximate any arbitrary relationship. So here we have input to output. So input will be the time and input as well as the state variable from the predict from the original mechanistic model, and the output will be W. So if you're giving any condition and you simulate the X variable from the original ordinary differential equation model, then we can predict W at that time. Then we can plug into a hybrid model to simulate the accurate model prediction. So these are the summary of how I construct the ANN. So these are the summary of how I construct hybrid model. First, again, from available measurement, we estimate W. After estimate W, we're trying to generalize through constructing ANN. And based on ANN, we can plug into original ordinary differential equation model. Then overall, this hybrid model can be used to predict the dynamic under new condition for the system of interest. So I briefly go over our case study for NF-κB signal pathway. So I'm interested in the NF-κB signal pathway under the lipopolysaccharide, which is a common component in the gram-negative bacteria such as E. coli. It will induce inflammatory response by synthesizing TNF-alpha. This is well known, but this is the formulated proposed signal pathway. It's quite complex. We have a lot of biomarker involved. The other one is we are actually interested in how NF-κB signal pathway is induced through two different stimuli, lipopolysaccharide, as the other one is called blefaridine A. So blefaridine A is commonly used in experiment and also is known to induce the inflammatory response through NF-κB signal pathway. However, it's how it exactly induced the signal pathway is yet not is still be to elucidated, and how it co-simulate the NF-κB signal pathway along with IPF is still a mystery. So we want to use a hybrid model to predict the system dynamic. So we use the mouse macrophage to get the to measure the TNF-alpha and RKB-alpha dynamic. So we got the model from literature. And so these are the original model prediction, and you can see there's quite this big discrepancy between prediction and experiment measurement we have. So here, the blue dashed line represent the experiment measurement, the model prediction from original model, and the error bar represent the model measurement. We have a quite discrepancy. So this is how we get the hybrid model, and so these are the final result. So the blue dashed line represent the experiment measurement we have. The original model prediction we had before I showed before, and the point with the error bar represent the measurement we have. The red solid line represent the hybrid model. So overall, with the hybrid modeling, we can see that the overall prediction accuracy increased significantly compared to original model prediction, showing blue dashed line under different condition. So which validate our hybrid model approach. However, you can see that still there is some discrepancy under some condition shown here and here. So which need to be further addressed. So in this direction, we are currently trying to increase the dimension of W so that we can have a further accurate result in terms of prediction accuracy, because we only estimate W at the time point when only measurements are available and doing linear interpolation between those. So which may compromise our prediction accuracy. So we're trying to increase, one way is we increase dimension W, so increase the prediction accuracy. The other one is we can increase the fidelity of mechanistic model, so we can have a better mechanistic network to increase the overall prediction accuracy. So in summary, we propose the hybrid modeling approach to enhance the model prediction accuracy when you have a limited knowledge. So as in the end, I want to acknowledge my supervisor, Professor Joseph Kwon, and Texas University, as well as our collaborator, Professor Jaraman. With this, I want to thank everyone for attention, and please feel free to ask questions now or after. You can send me an email at this address. Thank you.
Thank you very much. We really appreciate your time. And if we could show Dongheon some love with a couple of emojis and claps, that would be great.
Thank you.
So next up, we have Bradley. So Bradley, whenever you are ready to begin screen sharing, that would be great.
All right. My talk today is going to be on the role of representation in biological modeling. And on the first slide here, you can see 3 representations of the same scene, and we'll talk about why that looks different in the context of modeling. So when considering a biological system, it is important to consider what is the most appropriate level of fidelity. We talked about that a little bit in the last talk, and by fidelity, I mean realism or detail. And we find that there's generally a trade-off between parsimony, or models with minimal detail, and high fidelity, models with maximal detail. So what level of fidelity and the type of detail is acceptable in a biological model? That's the basic question you might ask when you start modeling. And this is an example of a number of models that we can use for biological systems. We start with this sort of spherical cow in a vacuum, and we can move to whole organism models or very detailed models of a molecular pathway. And this trade-off is thought of in terms of the level of detail, which is the spatial resolution or number of components, and the vertical scale of organization, so from whole organism to very small molecular details. And another related issue is model realism. So model realism deals with how realistic is your model. And so on the left, you can see these models of frogs. You can see these models of airplanes. This is a real airplane. This is a model of an airplane. And this is a model of the inside of a C. elegans. If we were to look inside and look at the muscles and the other parts of the body wall of C. elegans, this is what it would look like. This is from a virtual model from the Open Worm Foundation. But you can also do this at the behavioral level. And this is an example of sort of a behavioral Turing test that shows one example of an actual worm being observed with a microscope and another example of a worm being simulated. And the question is, can you tell the difference? And so this is why model realism is important, because you want to be able to get these details in place, build models and run them, and have them be indistinguishable from the biological case. And so we go from the spherical adult or developmental phenotype in a vacuum to hyperrealism. But maybe that's not what we want. Maybe we want something like a black box model. And, fidelity isn't always the most important thing. So there are a couple of references where they kind of grapple with this issue. The first one is this paper on physiological fidelity or model parsimony. And this is in toxicokinetic modeling. So in this case, and we'll see an opposite case in a minute, we cannot disregard physiological fidelity. Using simple models affect the steady state of estimates for chemical accumulation. So in this case, they're having they see this trade-off between parsimony and physiological or model fidelity. And so in their case, parsimony is this most simple model, this black box, and they see it is being in conflict with this idea of fidelity. The second paper is balancing physiology, anatomy, and immersion, how much biological fidelity is necessary in medical simulation. So medical simulations, they're trying to teach people how to interact with like a biological system, a human body, and they find that more detail in their models increases the amount of immersion you get, so you can learn more, and it generally serves to engage learners. But upon actually doing some testing with different types of models, they find that actually low fidelity models are probably adequate. And so maybe sometimes they're often preferable. So fidelity does give you more engagement, but it also is not necessarily always necessary to understand the system. So this is an issue of finding the right level of abstraction. So we're familiar with black box models where you take an input, you put it into this black box, there's this process you don't understand, and you get an output. But there are different ways you can model this black box, and you can add detail in later. So the question is, how much detail do you need to really understand a system? So we can take, say, artificial neurons or a simple perceptron, and we can understand basically how parts of the nervous system work. We can understand neural communication through this model. But we also want to know maybe the anatomy of a motor neuron, and that's certainly helpful in understanding the function of a motor neuron. Is it necessary? Maybe, but we can add this detail. But of course, if we take a model of the anatomy, we're also missing certain other aspects, such as ion channel activity. And so this tells you some other aspect of detail in the function of this. So all three of these models have different aspects of the function, and some parts of the function are black boxes. And so this is why, one of the reasons why fidelity is important, but sometimes we can just basically take one of these models and understand most of what we want out of the system. Sometimes, abstraction, you have this problem of overdetermination or underdetermination. So we've talked about underdetermination, where you take a black box and you can understand it using a minimal representation. But in this case, you have a chair where you're modeling it by looking at the material, by looking at the function, and then by looking at the particle physics of it. Now, the particle physics aren't necessary, but maybe they are in some cases. It depends on what we want to look at. So we want to refine these black boxes sometimes to white boxes. And so this is a process that they've talked about in the literature where you're taking these black boxes, you start off with something you know nothing about, and you try to find the function and assign function to these black boxes and then break them down. So that's one aspect. Another aspect of this is looking at levels of representation. So, progression model is what we call representation free. That means we don't have any sort of framework for understanding what we're looking at. We just take the data and we plot it out. In a representation rich model, by contrast, we build some sort of hierarchy or some sort of framework to understand the data that we have. And so that's also part of this trade-off. So again, you have this model of gene expression that outputs a set of quantitative values. That's more of a representation-free model. And a representation-rich model is where you actually have sequences and you're looking at this process of transcription and translation, and you're getting outputs that tell you something about that process and how it works. And so we've done this with what we call meta-brain models in my group, where we have these models where we layer them based on their level of representation. So you have a sensory input or an input of data. You have these representation-free models, which in this case is a connectionist system, a representation-rich model, which is a symbol manipulating system. And critically, they interact in different ways. So you can actually exchange like information you might get in a symbol manipulating system with the processing power of a connectionist system, and you get an answer that is sort of bridges those two. So it's a hybrid model, but it works in ways that sort of like how our nervous system works, but it allows us to take advantage of both of these attributes. And so we have a talk on this that was recently given, and there's a paper forthcoming on this. So finally, I want to talk about maybe how you look at a phenotype using a certain type of model. And in this case, we're using a certain type of maybe underdetermined model called a cellular automata. And so we know from just by observation that cellular automata can model something like a shell here of a marine invertebrate. And they have basically, it's generating the same pattern on the surface. But this is, the underlying processes aren't here because we are just observing the cellular automata, and it's this computational model that's generating patterns. But there have been observations that cellular automata models exhibit computational universality, which can simulate every possible state configuration. So in that way, it's universal, and maybe we can say that we can actually replicate this shell. We don't even need to know the genetic level of what's going on, but we just want to know about the pattern formation. So these models allow for interactions between discrete cells, the models on the left, and they can mimic this sort of complex biological patterning. But of course, we don't know anything about the genetics of the system, and maybe that's important for understanding this process, but we can generate them with this underdetermined model where we're just generating patterns. And it looks very similar to this. But I think, there's also a case to be made that the genetics are playing a role that we don't really appreciate, or maybe there's some control there that is not, that is unspecified. So these are all issues. This is, I think, an example of how to frame a lot of these issues. So thank you for your attention. And my group DevoWorm does a lot of work on these sort of developmental processes and other types of questions. So thank you.
Thank you very much, Bradley. Round of applause for Bradley. Thank you. All right. So next up, we have Megan. And Donalyn, would you mind switching the spotlighting?
Oh, sure I can.
If that's okay. My thing froze.
Yeah. There you go.
Awesome. Thank you so much. Megan, the floor is all yours.
Thank you. So today I'm going to talk about how I think about tumor heterogeneity or diversity. So typically when people think about cancer and diversity and heterogeneity within that tumor, they often think of genetic diversity. So what types of mutations does the tumor have and how does that vary across the tumor? Here's an example here where they use the Shannon index to kind of quantify that, and they see that individuals that have increased diversity have a worse prognosis. But a lot of my postdoc work, I work closely with Dr. Padron, who's a hematologist at Moffitt Cancer Center on CMML, which is an overlap syndrome. So it's characterized by bone marrow failures, large spleens, and things like that. There's actually no curative treatment for this disease. It is a disease of the stem cells. And what he sees clinically is that there's a lot of variability in how the patients present. A lot of them present with an incidental finding, which means they get a checkup and their blood values are off. And then it's kind of under investigation, they find out, oh, they had CMML and get referred to him. And from the mutational standpoint or that genomic standpoint, what we see is that the mutational burden is really low. So melanoma, which is kind of the hallmark for really high mutational burden, we can see is on the right side of this plot, has a lot of mutations per exome, but CMML is at the total opposite side. So there's really little genetic diversity. There's only a handful of mutations that actually describe this disease. So the question is how, kind of where's this diversity coming from, and can we find clinically useful ways of identifying that to stratify patients? And so our idea was that let's look at the transcriptome. So we use single cell RNA sequencing, and I applied a generalized diversity index from ecology to work on trying to identify how we can quantify this heterogeneity in clinically meaningful ways. So here's the generalized diversity index. We affectionately call them diversity curves. This is a neat little function. So Q is this order of diversity, which you can really think of as a resolution term. So it allows you to tune the weighting that you're going to give for richness versus evenness. So richness is kind of, you want to make sure that everything is accounted for in your system. So even if it's in a really small part of the population, it gets accounted for, versus evenness, then that kind of the dominant species take a bulk of that kind of weighting. And so the Shannon index, which I've already talked about, and the Simpson index are two that are really commonly used in ecology. Shannon kind of tries to equally weight them, and then Simpson is more of a dominant dominance metric. And so to kind of get our pipeline up and running, we actually looked at applying these to publicly available data sets. So there are 2 healthy bone marrow samples and 2 AML samples. And what we found is that across this queue, we always had higher diversity in these AML samples compared to healthy. And so to kind of see how robust this was, we did some kind of downsampling exercises where we threw out half the data, reclustered, and see how that affected the clustering. And what we saw was that the diversity score didn't change more than kind of 1 or 2 points. So if you were to shift down the AML curve and up the healthy curve, you still got some separation between these. So this seems like a fairly robust metric, which is exciting. Then we were trying to dig into finding out more about what is kind of some of the meanings that we can find out from this metric. And so one was looking at clonality. So kind of at the bottom, I show the traditional hematopoietic lineage. So you start with your stem cell, then you have progenitor cells, and then you have further differentiated cells down the way. And what we see is that the HOC, that more polyclonal type, that has the potential to become the most other type of cells, has higher diversity compared to those further differentiated states. And further, we were able to see these same diversity differences in this sample from lung cancer, where we had normal tissue from the cancer patient's lung, but then also different areas of the cancer tumor. And again, we see that same, the kind of normal healthy tissue had lower diversity compared to the tumor. And so a lot of my postdocs has actually applied this to CMML samples. So we recruited patients to kind of quantify diversity and try to find those markers of clinically meaningful biomarkers of diversity, but we can also extend this metric to other things. So kind of towards the end of my postdoc, I met a kidney surgeon, Dr. Manley, and we could actually use this diversity metric to quantify tumor immune infiltration from kidney cancer. And so we could actually use this from bulk RNA sequencing, which was really interesting. But this allowed us to have a much bigger cohort. And so we had a kind of new challenge of previously I showed these kind of nice diversity curves and you looked at 4 or 5 of them, and those are very kind of interpretable and handleable. But with the bulk RNA sequencing, that's much easier and kind of cheaper to run. So we had cohorts with over 100 patients. And so now it was kind of a little bit challenging to look at that spaghetti plot and kind of interpret some of what is going on. And so we actually could distill these curves down to what we're calling these point estimates of diversity. And in reality, we can actually get down to 2. So here we have kind of 5, but really what we get are richness versus evenness metrics. And so kind of our low Q diversity, the inflection point slope, and our delta D are all kind of measures of richness. So again, you're giving more weight to all the different types of species around. And then the high Q and the inflection point Q are actually measures of evenness. And so, and distilling it down to kind of 2 numbers is easier to compare against these really large cohorts. So that's kind of what we've transitioned to kind of moving forward. And what we've seen is really interesting and probably not super surprising, but the patients that had kind of larger tumors, higher grade, had more immune infiltration compared to tumors that were smaller. There's this sarcomatoid status, which is a kind of a more aggressive phenotype that also had more immune infiltration. And we saw that in both T cell and B cell receptors. But this was not really informative with respect to survival. What we found is that that inflection point, so that measure of evenness, was actually a way we could stratify patients based on survival. So we have, if you had that, if your inflection point was kind of more to the right and kind of larger, that's kind of the larger or higher diversity value, you had an improved survival compared to if your inflection point was more to the left. And so we found that this GDI metric is really informative way to quantify intertumor heterogeneity, and we can actually use it to stratify patients. But like we just kind of saw in the kidney cancer example, these different point estimates have different meanings and how you would want to interpret it. And so kind of to end on some of the kind of next opening questions are really, how does this change over time? When we get these samples from patients, whether it be from single cell RNA-seq or bulk RNA-seq or a variety of other methods, it's kind of a snapshot. And so the question is, how does this change over time? And we have a little bit of evidence. This is a paper that looked at breast cancer cell fusion events. And we can see that over time, you get kind of this influx or this increase in that diversity value. So this kind of teal curve right here is this passage 2. And so the parental lines are this kind of reddish and this purple line. You see a spike of the curve go up, and then it kind of comes back down between the other 2 for the green curve. So those are kind of the open questions and some of the next steps. Again, a lot of this work was actually done for my postdoc, so I want to thank all of our collaborators, our funders, and our patients. And I'm happy to take any questions in the breakout room.
Hey, thank you so much, Megan. Thank you, thank you. So if we could share a few emojis and clap for her, that would be great. Okay, so in a moment, we are going to open up breakout rooms, and you will have the opportunity to navigate between these. So in a moment, what I'll do is share my screen so that you could see what we mean. So here you will see all the different presenters. And for those that were here yesterday, you could see that you just join who you want to have more conversations with. So all you need to do is just go to the room that you'd like to start having conversations in, and then when you're done there, you can navigate to the next room. So all you need to do is just click this blue join button right here, and that will get you to the presenter. Donalyn?
They find that by selecting the breakouts at the bottom of the page, right?
Yes,
Awesome.
great point. Okay.
I can also.
And if you're having trouble and you want to tell us where you'd like to go in the chat, please let us know and we will manually put you in the room. So just let us know via chat where you would like to go and we'll manually get you there.
I think people are figuring it out.
Awesome. So I see Alicia, Angel, Likun, Cece. And again, you all have time to be able to navigate between presentations. We have until 10:30. So you don't have to just choose one and stay in one the entire time. You can take some time to visit each of the presenters' rooms. Okay.
They're
Go ahead.
starting. I see LeQuinn just moved. They're
Okay,
on
awesome.
their way. So we have Alicia, Sissy, and Angel. Oh. One of them just left. Angel left. Alicia and Sissy, are you here? If you